This session is on cancer biology, and we have 3 exciting speakers lined up. So I'm looking forward to the talks. I'm looking forward to the very lively Q&A session that we had in the morning to continue. So without much further ado, Dr. Karthik Raman from IDM.
Okay, thanks a lot. It's really a pleasure to be back at ISC. And as I was remarking to a few people, this is the very hall where I defended my thesis, so all the more fond memories for me. And that was the last time I presented here 14 years ago. So today I'm going to talk about decoding cancer driver genes. I'll tell you a little bit more about it as we go on. So I think everybody is now very familiar with cancer genomics, but essentially cancer is a disease of the genome. And we have various kinds of alterations that you find in cancer genomes. There are point mutations, structural alterations, and so on. And I think there are a litany of genomic studies that have given lots of very interesting insights into the makeup of cancer genomes. And one bunch of studies have focused on what are the driver and passenger mutations in cancer genomes and how one can use the knowledge on these to understand the etiology of cancer. So what are driver mutations? These are those that directly or indirectly confer a selective growth advantage to the cells where they occur. And passenger mutations are just passengers. They don't seem to have any known connotation of that fact, so they don't seem to affect the progression of cancer directly. And if you see, there are so many interesting studies. I'm just going to quickly show you a collage just so that you get convinced that there's enough work going on here. And I'm sure some of you would have read a few of these papers at least.
Hey, sorry, Karthik, I think they need you to mic up.
Oh, so that it can be recorded.
Thanks.
Am I audible? It's on. Yeah. Okay. So these are a bunch of tools that have been already developed for identifying driver genes, and some of these papers are particularly interesting because they've tried to uncover what are the, using essentially simple statistical methods, very key signatures of various kinds of cancers. For example, do smokers exhibit very specific mutational signatures and so on. So this is like a very, very classic paper in this field. With it, the methods for identifying driver genes can be broadly bifurcated into two. One set of methods based on what is known as background mutation rate. So essentially a statistical method. You just count what is the number of samples in which a particular mutation occurs. So if it is significantly higher than a particular threshold, it's a very oft repeated mutation. So you want to then conclude that it's a driver mutation. The other set of methods essentially try to classify mutations into different types based on functional impact. It uses ratios of various kinds of features. So it's essentially a machine learning driven approach wherein you pick various features, and I'll talk about some of this tomorrow, right, in grand challenges where can you actually explain why these things work, does it make sense to use these as features and so on. But the point is they're able to identify these driver mutations with accuracy and not relying on just high mutation rates. So you may have rare mutation events which are still drivers and you want to capture them. So this is a very important aspect of what are known as ratiometric approaches. And if you see, this just tries to give you a very loose picture here. So if you see, the ratio of silent to missense to loss of function is different for different kinds of genes. So what is a neutral gene versus a tumor suppressor gene and an oncogene. So I'm going to use these terms. Don't worry too much about the biology underlying it, but suffices to say that there are different types of genes. So there are those that function by suppressing tumors, and when they lose the function, they contribute to tumor. And there are those that acquire oncogenic function, and so they have a gain of function to actually result in tumor progression and so on. So what are the challenges? There are a large number of driver genes that are still to be identified, and many of them have very low mutation rates. So you really need to sift them out of, you don't have like an easy background to pull them out of, right? When you have a very high mutation rate with respect to background, it's easier to pull them off. And you have heterogeneity across samples. So every cancer is very different, and even in the same person, you will see that their etiologies of their cancers are remarkably different and so on. And if most of the existing tools have very high precision and low recall, and you also see that except for a few well studied cancers, you really have a paucity of samples to deal with, and let's not even talk about Indian samples and so on. So we actually are fighting with very few samples and still try to make inferences. And if you look at these classic plots, right, so this tells you what is the mutation frequency, and your famous genes like BRCA1 show up here. But there are a lot of mutations that have very rare frequency below backgrounds. So you really don't have easy methods to fish these out from data currently. And if you see even from the last bench, you can see that this is a very dark figure, which means that the values are very low. So you see that the recall is very low, even though precision is reasonable for many of these methods. So clearly there is a lot of room for improvement in these methods, and there is room for one more method. Right? So our question is, how do we reliably predict driver genes in cancer genomes? And I'm going to talk you through a few methods that we've developed. So there is a method called CTAG, then a method called pivot, and if I have time, I'll briefly talk about this pipeline and this method as well. Just a couple of slides on each of these, but I'm going to focus my talk mostly around this particular piece of work today. So how do we predict driver genes in cancer genomes? And the key objectives for us are to identify important features and of course avoid overfitting, because you just don't want to learn the pattern in the data all too well. And ultimately identify novel driver genes and validate them against literature. So we go mostly with coding data, and we define a bunch of features. So there are some features that folks have previously defined, but we've added new features to that. And we built a standard train test model. We use random forests and so on. I'll not get into all those details. But just look at these features. So high function impact by low function impact, high function impact by benign, number of medium functional impact mutations per kilobase of the gene, and so on. So if you see, there are these litany of features that one can define, and many of these seem to have a significant impact on how it impacts on whether it's how it impacts driverness, so to speak. Right? So are these features going to be important in determining whether your gene is a driver or not? And remember, we are comparing against just background mutation based genes. And if you see, our performance was reasonable. We got some very good values, especially for predicting tumor suppressor genes. But if you see in the case of oncogenes, the values are a little poorer. And we suspect that a lot of existing models have done a lot of overfitting. And I mean, I can't say this with certainty, but looks like all the experiments that we ran, we could see that a lot of existing models were indeed showing quite a bit of overfitting. And if you see that these were some of the new features that we identified. So this is just to tell you where just start thinking about how there may be a functional impact in some of these kinds of mutations, and how it translates to something actually becoming a driver. And these distribution plots just show that there is much more similarity between what we see in the test set versus what you see in the data. Okay. Okay. So our feature contribution shows that loss of function and missense features captured a lot of differences between the TSGs and OGs. And if you see, some of our predicted tumor suppressor genes are already known driver genes, and many of our top predicted oncogenes are also known driver genes. And this is basically validation against literature. This is what we've done. We've obviously not done experimental validation of any of our driver genes thus far. And more genes for people to look at, for the biologists to look at. But I think overall the picture is that there are ways to carefully sift through the data on mutations. I didn't actually go through the data. We have like millions of mutations drawn from Cosmic and so on, and actually computed various features for these, and ultimately make reliable predictions on what is a potential driver genes. And if you see, Mute6CV is one of the top tools that currently exists, And if you see it, there is quite a lot of divergence between what you see in the predictions of MuteSig and what is actually told in the training data even. So there is quite a bit of deviation. That's because of the bias. Undoubtedly, it's a great method, but there's still a lot of bias towards background mutation rates in existing methods. Okay, but okay, with this, let me just move on. I think I'll skip some of this. Essentially, we identify new pan-cancer and tissue-specific driver genes, and lots of tissue-specific predictions also were made. But the big question is that tumors are very heterogeneous. And can you identify driver genes in an individual person? Meaning, the same gene might be a tumor suppressor gene in one individual, and it could be an oncogene in another individual having the same cancer. Or it could be in the same person with different cancers and so on. So how do you actually carefully identify the role of these genes in the development of cancer? So this brings us to our method called PIVOT, which talks about personalized identification of driver oncogenes and tumor suppressor genes. Same old story, there are lots of rare driver genes are missed. So the assumption is that there is always a driver gene in almost all of these cancers, and if you see, in many of these cancers, there are no detected driver genes at all. So it means that you really need to cast the net wider, deeper, whatever, to identify those rare signals which correspond to tumor suppressor genes and oncogenes in these data sets as well. And there is a lack of ground truth as well, and a lot of the methods just use unsupervised methods to try and rank genes based on their importance and so on. We took a different approach here. We used a supervised learning method here. And importantly, we also used multiple kinds of omic data. So we used not just SNVs, we also looked at gene expression data, we looked at network data and so on. And also, differentially expressed genes, not just based on cohort, but on individual samples and so on. So we used a lot more data than is normally used in these studies. So, so we labeled genes as driver or tumor suppressor genes or oncogenes based on multiple studies, which have large-scale studies which have tried to classify these genes. And we used quite a bit of data. We used SNV data, RNA data, somatic mutations, oncodomains, and also network data and so on, and a bunch of different models. So what we ultimately did was, we saw that mutation-based categorical features are not sufficient to predict driver genes. So a lot of the methods have been using, and even we did in the previous study, we focused on just using mutation DNA data. We could see that RNA-based features were able to help predict personalized driver genes, and we had, in fact, an accuracy of greater than 96% across the best models for 3 different cancer types that we studied here. And network-based features were also coming out as quite important. So the moral of the story is that you cannot just go ahead with simple DNA data. It sounds obvious, but we could really systematically show this here, that you need to not only use DNA data, but if you have RNA or network data, might as well leverage it to make more accurate predictions. So ultimately, we had a multi-omic model with about 99% accuracy across the best models for the 3 different cancer types. And this Bailey et al. Is a classic study that appeared in Cell in 2018, which looks at labeling individual driver events based on a lot of information. They have structural information and so on and so forth. So these were our key learnings, and again a big map of numbers for you. But if you see that there are the numbers for Bailey et al. Are consistently good across different scenarios, like either we are looking at neutral genes or oncogenes or tumor suppressor genes. But beyond all of this, I think what is important is to understand that you can now look at these in a very cancer specific fashion. And if you see, these are, this is for breast cancer, this is for colon, this is for lung adenocarcinoma. And what you see is that many of the top genes identified have already been flagged as important driver genes. And I think for some of these that are ranking high but still not been identified in specific individual studies, we can really look a lot closer at the biology of these. And this is something very interesting I would like to point out. This particular gene called JAK1, I think some of you may be familiar with it, but it has been identified as a tumor suppressor gene in breast cancer and an oncogene in colon cancer. And current methods cannot identify these dual functions of a single gene based on the data. So I'll not get into all of the biology of this, but there are so many different studies that have shown that this particular gene has diverging roles in different cancers. And this is something that we were able to again pull out based on, by essentially doing a bit more of an unbiased study, not focusing on just the DNA mutations, or by integrating multi-omic data, and also not focusing on the background mutation rates and so on. So to summarize, Pivot predicts driver genes in a single patient using multi-omic data, and the altered genes are labeled as tumor suppressors or oncogenes or neutral. And the very same gene can be labeled as TSG in one patient and OG in another patient of even the same cancer type, because all of these predictions are based on a per sample basis. And the list of rare predicted genes provides a nice bunch of genes to target and study, because a lot of the drugs that are today being developed for cancer are focused on these driver genes. So it makes a lot of sense to have different methods throw up driver genes, and obviously you need more validation and filtering and so on, but I think this is a very good place to start if you want to look at personalized medicine and better therapies and so on. And ours is the first supervised ML model to label genes in individual patients, and essentially reaffirms what biologists have been saying for long, that genes can behave differently based on the signals that they receive, based on the environment and so on, and it's up to tease out, it's up to us to tease out this information from the data. Okay, so I think I'm doing good for time. So I have a couple more slides on a very interesting study. So this is this essentially came out of a very interesting hypothesis that we once discussed with Professor Ashok, which is whether sequence neighborhoods can actually flag a particular mutation as pathogenic or not. So this is very interesting. So this is your regular gene, ATCG, whatever, and there is a particular mutation here. So this mutation, it could be either pathogenic or not, meaning it could be a driver gene mutation or not. Can you predict its driverness by simply looking at its nucleotide sequence in the neighborhood? Okay, it turns out that, so we started with the question of, is there a difference between the neighborhoods of pathogenic and non-pathogenic mutations? And it turns out that not only is there a difference, you can actually use that difference to even make predictions, which is what we did. And it turns out that I'll cut to the chase. So we looked at different neighborhoods and extracted features, except for the neighborhood of plus or minus 1, which is very trivial. We could see that there was a signal in all other cases. So we did variations from, I think, 2 to 10, if I remember right. So you can make predictions of whether a particular mutation is pathogenic or not by simply looking at its neighborhood. So we so we got very good results. For example, our sensitivity was as high as 0.938, and we also compared it with other methods and integrated it with other methods and so on. And this is particularly useful at predicting unseen pathogenic single nucleotide polymorphisms. So this is something that again, you can use this as one of the methods that you throw into the mix to make these predictions. We can always have other levels of filtering, but this is again a very potent method to identify driver mutations from data sets in a very unbiased fashion. And ultimately, we thought we'll put all these tools together for people to use it. So we have this very simple GUI based tool that people can use for extracting insights from whatever data sets that they have. So it uses all your standard DNA sequence analysis, WGS methodologies, and also for RNA-seq, various kinds of aligners, differential expression tools, and so on. So here's the array of tools that you can use. So you can either use the tools out of the box, or you can tune the parameters for each and every tool. So depending upon your level of comfort, but there are some things that work just out of the box. And this is how it looks, very simple interface. Just dump a folder of samples and click and run. That's all there is. So that brings me to the end of my talk. So to summarize, I did show you a bunch of tools for predicting driver genes and passenger genes from genomic data. But many of these tools have very low recall. And I hope I've convinced you that these ratiometric approaches as against background mutation rate approaches are much more powerful for predicting driver genes. We had a pan-cancer model called CTAG, which identifies this in a tissue-specific fashion based on 19 new features that we had engineered. But the point I was trying to tell you is that the tumors are highly heterogeneous, which means that you have so much variation between different cancer types and across patients, and even within patients who have the same cancer type. So can you still identify these in a robust fashion? And given that current methods only rank genes as possible drivers and there are no supervised ML methods currently existing. So Pivot predicts driver genes using multi-omic data. We use DNA, RNA, network data and so on, and is able to capture heterogeneity well. Essentially, as I'm probably repeating myself, that the same gene can be labeled as TSG in one patient and OG in another patient of even the same cancer type. And the last tool I talked to you about, NB driver, essentially shows that mutations have a very definite context and you can actually exploit these sequence neighborhoods to make predictions. And if you want to try any of these tools, you're welcome. You can use this GUI tool that we have developed to tinker around. I think I'll skip this. So I'd like to acknowledge quite a few people who've been involved with this. So this has been a large team effort over the years, but spearheaded by Malvika Sudhakar, who's now pursuing a postdoc in Denmark, and also with Professor Raghu from chemical engineering. Shayantan was involved with the NB driver study along with Professor Ravindran from the computer science department, and these are the folks that were involved with the iComic pipeline generation. So I'd also like to thank the Department of Biotechnology and IBSC and RBCDSI for funding, and thank you for your attention, and I'm very happy to take questions.
Which machine learning models do you use?
So in this case, we used random forests.
Okay.
Yeah, but we've used different kinds of machine learning models as well.
Okay. And the other question is, you talked about multi-omic data. Is there a data integration step also required there, or?
Fair point, but we basically extract features, so the integration is sort of automatic. And we basically extract features from DNA data, features from RNA data, and features from network data. So all these are fed in as individual features to the model, and essentially that's the integration step.
Okay, sure. Thanks.
I have a question, which is, I think last year Professor Deborah Marks came up with a paper on identifying pathogenic mutations of all variety using some deep learning based method. What I understand from your talk, although you didn't really talk specifics about it, so you are engineering features. That means you are mindfully constructing the features. For example, in the case of where you are learning from the nucleotide context. So what sort of features are you?
So in the nucleotide context, the features are very simple. It's basically a one-hot encoding of just the windows, right? So if there is an ATT around this particular mutation or a TTT around this mutation and so on. But in other cases, we actually look at the ratios of various kinds of mutations. There is also this tool called polyphene that we use to predict the functional impact of any given mutation. So suppose there is an A to T that's happening somewhere. What is its functional impact? Is it
SIFT and polyphene, all this methods?
Exactly, right? Okay. So we use that to figure out what is the ratio, what is the fraction of benign mutations to high functional impact mutations and so on. So these ratios of mutations apparently have a big effect on how it turns out for the gene, whether it's actually a tumor suppressor or not, and so on.
So the final question is, are you also learning about which features are influencing your decision making?
Yes, yes. But as I was saying, it's still not satisfactory. We still can't explain it at a level that we would like to.
So I have one question. So in disease prediction, we will generally have, the disease population would be less and the healthy will be more. So we will face the unbalanced data set in this case. So how about when we do the oversampling technique? Will it?
Yeah, in this case we
so
didn't have to
maybe-
do that, but in there are many scenarios where we've had to, whenever we work with other disease clinical data sets and so on, we've had to use SMOT and various other methods for oversampling and
Will it
things
cause
like
some
that.
sort of overfitting in the data? Like
No, not overfitting as such, but no, not overfitting. So I think there are standard methods that have been developed to counter for these imbalances in the data. So we've also found that multiple times while looking at, say, predicting essential genes, right? So if you see only 10-20% of genes in an organism are essential. So majority of examples are of healthy, of non-essential genes. So again, you have to resort to some of these methods, and I think each method will have a few, which is why it's important to have like a multi-model based approach to make these predictions.
yeah, because SMOT might increase the disease patient and it might give a very high precision and F1 score.
Possibly.
So in that case, I might be.
Questions? Let's thank Karthik for a very interesting Our next speaker is Dr. Shabrinathan Radhakrishnan from NCBS, who is finding his way to the podium. Thanks, Karthik.
Doug and other organizers for this opportunity. Myself, Sabari. I started my research group at NCBS 4 years back, and our lab is broadly interested in understanding cancer evolution through genomics. As we have heard from first speaker and also from Karthik before, we know a lot about cancer. We know how it develops and how it progresses, and we know about the treatment options as well. But still, we cannot completely cure the cancer. One of the problems is it may either reoccur or it will develop resistance to the treatment. Why it is happening? Most of the time it happens because it continuously evolves. And the evolution is being fueled by genetic and epigenetic changes that is constantly happening in the cancer